recording it. Okay. Thanks for joining us. This afternoon or this morning, depending where you are, we're just going to quickly run through some of the new features in ioGAS 6.3. ioGAS was released just a few weeks ago, and if you don't already have it, then please feel free to go onto our website and download it. During the course of the next half hour or so, I'm just going to jump between a PowerPoint and ioGAS itself, so it's not a static display. Now there's quite a lot of new features in ioGAS 6.3. Some of the headline features are listed there in the PowerPoint, and the top 3/4 or so of those will actually dive into ioGAS and have a look. But there's a myriad of other minor improvements as well, which we'll talk about on the way through. But the main ones we'll cover off are the connection of ioGAS direct to the MDEX Hub IQ database. A lot of the tools that we have out in the field generate data and can be connected to our hub. And if you want to get that data back down to ioGAS to work with it, then you can get it in direct from the MDEX Hub. We'll look at importing random data and taking random subsets, some enhancements to the StereoNet tool, distance plots for a new method of identifying outliers, and then have a look at biplots, which is a variant of PCA, variable importance, and a new robustness algorithm. Just before we get going, a couple of immediate things to notice about the interface, which you'll see when we're using GAS. There's now a memory meter in GAS down the bottom right-hand side, a small green bar. So if you're working with your data, especially larger data sets you've been working for quite a while, and you start to notice that the green bar is getting over towards the right, then you should really just save your data and jump out and then load a higher number build of ioGAS, which will take up more memory and allow you to keep working with those larger data sets. The edit menu, which was in GAS up to version 6.2, has now been removed, but what was on the menu is still in GAS. We've just moved the remove invisible columns and remove invisible rows and remove columns entries under the data menu, and we've moved a couple of the other tools into the remove columns dialog. So if you have a look at it in GAS 6.3, there's now a series of new buttons down in the middle there. The top one there, that allows you to remove all derived variables. The middle one allows you to derive variables which are functions of another variable, so just taking up room in the file for no reason. So if you've had conversions of element to oxides and units and other things that have just crept into the data file that have a correlation of 1 with another piece of data in the file, then you can get rid of it. And the bottom one there is just allows you to get rid of columns that actually have no data in them at all, which sometimes happens when you're importing data into GAS. Now, first things first, I'm just going to jump into GAS now and show how you can connect through to the MDEX Hub or the Reflex Hub IQ database system direct from the menu in GAS. So this is GAS, new version. There's the memory meter down on the bottom right there. If you keep an eye there, you'll see that moving as we're bringing data in and out of GAS and closing and opening files. Now, if you're using our equipment in the field, you'll have a Reflex Hub or an MDEX Hub IQ login. It's those same credentials which you add into GAS, which go into here. So there's a username and password. So you're supplied with that when you rent the equipment. So I'm just going to hit OK. And now GAS is actually going into the MDEX Hub now, and what it's doing is it's going to bring down data which is available via searches which you've saved. So you could have set up a search back in the main online system for data that you access all the time. If you save that search, then you'll be able to come in GAS and go to it directly. So in this case here, I was going to pull down some gamma data which has been generated by one of our easy gamma tools. So there's no import export going on here. GAS via the internet's gone and logged into the MDEX Hub, and it's dragging that data across the net right now. Says. So across it comes. and here's the dialog. So it's brought down project, drill hole data, depth, and the gamma. The rest of the data there is metadata associated with it. I can just click OK. And then go and pick the gamma data. So there's the gamma there. And if I want to make a downhole plot of that, I can go to the downhole plot tool. Just pick the drill hole, and because it's point data, we put the same depth column in from and to, and OK. Select the drill hole, and there's the gamma data plotted up. If you had multiple holes there, it would bring them all down in the project at the same time, and then you would get a series of gamma plots across the screen. There are other data types that are available as well via the Index Hub. So another one, for example, I could go back in and just say I don't want to save that at the moment. I'm going to go back in and pick out some XRF data, for example. So if you're using our Reflex XRF tools in the field, they upload their data into the hub as well. So you could be running a tool remotely out in the field, be sitting back in the office and looking at the data as the data is being collected. This is the data that's been fetched over the internet. Those of you who are familiar with portable XRF and know the problems of text values and a lot of numbers and nulls and negatives and less than symbols, all those sorts of things. We bring it across in very consistent format, and here I'm just going to apply typical detection limits associated with the professional model of the Olympus machine that we use, and click OK. Just yes to that. And here's the data that's been brought through, and all of the data is all ready to go. It's all been aliased, it's all ready to go in GAS. So I can click OK again, and then pick out some elements. And there's all of the data. So all of the detection limits replacement work's been done, so you don't run into problems with nulls and zeros and things contaminating your data when you have problems when you try and log transform and do cross plots or scatter plot matrices like that. So I was going to go back into the PowerPoint now. So that was looking at the hub menu. The next thing I want to have a look at is there's two different ways of taking random subsets of data in GAS, and they're for two different reasons. Now the first one, let's go into Excel. I have a file here, so these days people bring data into GAS which aren't just sample level data, so they're just not analyses of samples being drill samples or surface samples. Often people are bringing derived data into GAS, and one of the use cases here is estimated data out of block models, which can be extremely voluminous. And in fact, we've had people writing to us wanting to import actually tens of millions of rows into GAS. Now GAS doesn't handle tens of millions of rows particularly well. If you're trying to make a scatter plot, I don't know any software that does actually. But nevertheless, it's a use case which people are coming up against. So. And if you're dealing with such voluminous data, then what you should be able to do is actually randomly sample it and plot the data to look for relationships without needing to plot all of it. If you've got 5 million rows in a data file, looking at 5 million rows on a 3" by 3" scatter plot on a screen isn't necessarily going to tell you very much, but if you could take a random subset of that data and plot it up, it would be just as valid a thing to do. Now here's a data file on my laptop. It's got 100,000 rows and 10 variables, so it has 1 million cells in it. It's not a very big data file, but nevertheless it demonstrates what I'm about to do. So if I scroll down to the bottom there, oops, out of the way. You can see there it's got 100,000 rows in it. Now I'm just going to hide that. Now if I go into GAS, go File, Open, I won't save that. Back into 6.3, and it's not a GAS file because I've not brought it into GAS already. It's sitting there as a CSV. Oops, I don't want to open that file, so I want to go into Import Random Subsample. So don't save there. Point it at this CSV, which has a lot of the data in it. And I can tell GAS to go and randomly take a certain number of rows or a certain percentage of the data. If it's a certain percentage of the data, tell it exactly a percentage. Now, if you give it an exact percentage, ioGAS will actually read look through the whole file, work out exactly how many samples are in it, and then determine exactly how many it needs to make a random selection from the total file. If you've got a really large data file, like millions of records, then don't tick the exact percentage, just put in percentage of the data, and GAS can determine on the fly then, without needing to read everything, how to get its random subsample out. This file's not too big, so I'll just leave it at 10. If you want to repeat this process later on, then you can always write down the random seed, because if you use the same seed, you'll get the same samples picked at random. So here I go, let's just go OK. So it's gone and looked at that file, and if I don't need to go any further here, but if I look at this dialog, it says it's pulled in 10,000 of the 100,000 rows, and it's made a random subset of them for you. So that's one use case. I'm just going to bail out of that now. There's a second use case. I'm just going to open a GAS file so I can show you that. Now in this case you may have loaded a data file and you might be looking at some relationship in the data, but you might be wondering if that relationship holds up when you're looking at a subset of your data. So often, excuse me, if we're looking at larger data files, if we look at smaller parts of the data, then sometimes the relationship doesn't hold up quite as well. So there's a way to test that in GAS, and that's to use tool under data again. So here we've got generate a random subsample. So this is different to the previous tool. This time we're going to generate random subsamples of data that we're already having GAS and we're already looking at. So I'm going to set that at quite a low percentage, say 10%. And now I'm going to subsample this data. At the same time, I've got a spatial view of the data, and I'm looking at a regression line of iron against cobalt, both logged. But these axes are logged, so the axes aren't going to move around when I randomly sample the data. So I'm just going to generate it, and so here I've taken a 10%, a random 10%, so this is sampling with replacement. We've still got a good spatial coverage, but the relationship here still seems to hold well. But if I do it again, and again, and again, you can see here I'm taking random subsamples of 10% of the total data, sampling with replacement, and just seeing if it's affecting the regression that I've measured using all of the data. That regression line you can see on the XY plot's actually live. You can see it's barely moving. So you would surmise from that actually the relationship between cobalt and iron across this data set is pretty robust, because when you go and look at much smaller samples of the total data, it holds up, and in fact it barely changes right across that map area. So two different uses for random subsampling. Now back into the PowerPoint. So next feature we'll have a quick look at is the stereo nets. Now stereo nets were released in ioGAS version 6.2. Just let me get up to number 3. There we go. Let's open this file up and get that out of the way. So there's a stereo net of poles to the planes. This is a spatial representation in drill holes of where the structural measurements were taken. So I've just got to get the control screen out of the way for GoToMeeting. It's over the stupid thing. Let's move that down, out of the way. Okay. So stereo nets were released in 6.2. In 6.3, there's some additional controls over on the right-hand side so that we can show point density images. So it's the point density of the poles. As we click through, it just changes the opacity of the image. There are some controls we can apply to the point density as well. You can change the resolution of the image that's plotted over the top of the stereo net. We can change, if you don't want these blue areas drawn out to where there's very little data, you can change the threshold value in there like that. All of the normal GAS colouring bars are available as well, so if you wanted to show the data like that you could. I'll just close that and then go back to the opacity settings so you can see the points through it. So that was a very often requested feature with the stereo nets. Let's turn that off again now. And the other feature we have here is the ability to contour the data as well. I'll sort of open up the contour. Yeah. So you can see the contours in there. So you can adjust the number of contours, you can adjust how many cells should be in a region before the contour gets drawn around it, and you can also fill the contours so they look like that. I'll just then make that, whoops, that's the point density, wrong tool, and make them visible. The last thing you can do with these contours is that you can colour the points with where they plot within these contours. So the colour of these contour levels here are coloured in. You actually colour the points with where they are. And to do that, to give the focus back to the stereo net, I can just press this bottom button over on the right. So if I press that, you can see here that the spatial locations are now coloured with where the samples plot with respect to their point density on the stereo net. So they're just a couple of enhancements. There's a few other detailed things now which are available in there as well. As usual, as we go through here, we always recommend that you make use of the help file. The help file is extensive, and everything that I'm showing you through this demonstration very quickly is well described in there. Back to the PowerPoint. Now we've upgraded the way, so we've had two different methods of calculating Mahalanobis distances in gas in the past, the usual method or non-robust method, and a robust method which we call the M or S estimation. We now have a new method which we call MCD for minimum covariance determinant. If you're interested in it, that is the paper to read, which is there in the PowerPoint. It's also stated in the help file if you ever go and look in there. The best way to illustrate what we're doing with that is actually to show you in gas visually. So I'm going to go into another file. I think we're up to number 4. Let's get rid of that. So this is a couple of variables we're looking at. This is actually wine compositional data. So we're looking at malic acid versus proline. This is a series of chemical measurements of wine which were taken to determine if the wine cultivar could be identified. Anyway, but what it does provide for us is a good example of how the Mahalanobis distance is calculated. So it's just a scatter plot tool we're looking at here. Over on the right-hand side toolbar, you may never have interacted with these before, but these give you the ability to draw a Mahalanobis distance ellipse onto the data. So I'm just going to use the non-robust version here and click it again, and click it again. So that's the 0.975 probability contour for the Mahalanobis distances as calculated for all of that data in a robust way. I'm just going to do it again using a robust calculation because that's the best way to illustrate the difference between the two. So if I click this one here, and again, right. So there's two ellipses now. There's the non-robust and the robust. The robust is making use of the new MCD algorithm. You can see here that what we're looking at, and from a geochemical point of view, this is important, that when you have outliers, the outliers themselves always distort the analysis, and statisticians call that effect masking and swamping. So if we're looking at Mahalanobis distances to identify outliers, so samples are plotted outside the ellipse, you can see if you don't use the robust version, the presence of the outlying samples themselves distorts the ellipse, and in fact it tries to hide them within it. Whereas if we use the robust estimation of the Mahalanobis distance, we can see here that it much better describes the distribution of the data, and it more easily reveals the presence of all of these outliers. So that's how they work, and to make use of that in gas, we have a new type of plot called a distance plot, and you get at it via the Compute Mahalanobis Distance menu. Whoops, there's the entry there, so I'm just going to click it. Just ignore those for now. Say yes and yes and yes and yes. And there's the plot. So what this is doing is it's actually plotting the usual Mahalanobis distances, so the ones based on this ellipse, with the robust distances, so the ones based on this ellipse here. So you can see is that the samples that have low distances for both the robust and the usual solution are the ones that plot in there. And they're the ones that plot at that quadrant on the scatter plot. The ones that plot up in the top right quadrant have a high distance with respect to both the usual and the robust ellipse. That's those samples there. And the ones that plot in this quadrant are samples that have a large robust distance, those ones in here, but not a large robust distance when you look at the usual solution. So it gives you quite a lot of diagnostic information when you're hunting for outliers. And if this was geochemical data, it was a spatial view, you can colour the samples with the quadrant that they plot there and then go and inspect them in a map view. So it's just another way of trying to tag multivariate outliers in your data. That's that one. Now we'll just go back to a couple of tools that have been in ioGAS for a while, the principal components analysis and the discriminant projections, just to have a look at some enhancements of those. Just going to jump into another file. 1, 2, 3, 4, 5. Let's open up the data. So this is a series of Gaussian analysis, or Gaussians that were taken from different types of mineralisation in the New England Fold Belt on the east coast of Australia, which were analysed for a series of elements. So that's the data file in there. So I'll just minimise that. Now what I'm going to do now is show you a new enhancement to the principal components plot. Select some elements, those will do. Okay. So if I go into analysis, we'll go into the principal components analysis. Here are the entries that were present before, the classical and robust. There's a new entry here, so you can perform the robust analysis now using the new MCD procedure. But the other new entries here are biplot and 3D biplot. So I'm just going to kick off the 3D biplot, transform the variables in the Lawie. As usual, when working with all of these different data types in ioGAS, transformations are important. Let's click OK. So let the screen catch up. Is it keeping up, so the screen? Yeah. Now I'll just turn that. So a biplot is really, it's just a variant of principal components analysis. But what it's allowing us to do is to project the position of the elements and the position of the samples onto the same plot. And the simple way of interpreting these sorts of plots is that if samples that plot out along where the vectors are pointing are heavily influenced by those variables. So if I'm looking at that view of the data there, if I look at the classification over on the right here, those purple dots are from Kuroko-styled VHMS deposits. You can see that they have a strong loading on zinc, silver, gold and bismuth, which is as you'd expect. The Cypress type deposits are more heavily loaded down here on copper, moly and maybe iron as well. The epithermal deposits have a high barium and gold loading. So it's a neat way, because a principal components analysis is a projection using many variables at once. It's a very easy way to try and work out if you're seeing a grouping structure in your samples. When you've got the variables overlaid on the same plot, you can start to tell you what variables are important in creating that grouping structure in the data in the first place. So that's one enhancement. The other enhancement was the variable importance. So that's an enhancement to the PCA. I'm just going to close that now. Now, with the discriminant projection analysis, we've had that tool in ioGAS for a while. It's different to the PCA. The discriminant, whereas PCA maximises the variance that it can show in a single projection, discriminant projection analysis, I'll just open it up. If you have a priori knowledge of groups that samples belong in, is the best possible projection to separate the group means using all of the available input variables. So again, it's a linear model, or it's a linear projection. So here are the different groups, and here are the different groups, the known groups. You can see here using discriminant projection analysis, it can very well separate between those groups. Now, if we want to formalise that classification, we can use a tool which has also been in GAS for a while, which is the auto domain classification diagram. Let's click that and click that, and I'll just get rid of that one now. And now, rather than, geologists love classification diagrams, rather than manually creating it, we've used the maths here to go through and actually determine where to position those boundaries optimally. And that diagram is reusable, so you'll be able to classify new samples and project them onto that diagram automatically. The axes remember the calculations to place the sample in the correct space. Now, the additional bit of functionality we have now after doing this sort of work revolves around, well, I know that I can split my samples into different mineralisation styles. The next question you might ask would be, what are the most important variables or elements I've analysed for, if it's a geochemical example, for telling me which group is which or what group the sample belongs to. Now, to do that, I just need to go back and select the elements here, which I've done. So we go back to the input variables and under analysis, down to projection, discriminant projection analysis, and then to a new tool here called the variable importance plot. So I'll just open that. As usual, transformations are important, so we're going to tell it to log 10 on the way in. I'm going to tell it to do a full search. I'll explain that in a minute, and then let it run. And here's the answer. So what's happening here, the K along the bottom there, we call this a variable importance plot, just to give it an English name for geoscience users. Statisticians would call it all possible subsets analysis. So what it's doing, for example, where K equals 2, is it's looking into the data file and trying every possible combination of two of the inputs to see which two give the best group separation on a plot like that. Then it steps through and gets to 3 and tries every possible combination of 3, every possible combination of 4. I selected the extensive search when I ran that then, because there was only 10 or 11 sample variables in the file. It ran very quickly, but if you get up to 10, 25, 30 variables, it can really slow down quite a lot, because you imagine computation, it's very intensive. In that case there are some other search procedures you can use. So the interesting thing it's telling me there is that the most important element I included in analysis to allocate a sample to one of those groups was copper. And then if I look at the tabular report, it was copper, and then the most valuable pair of elements in that input was copper and manganese. The most valuable combination of 3 elements was silver, copper and manganese. So that's a quantitative assessment of what's important to allow you to make that decision. And intuitively, being a geochemist, you may not have included manganese in that group, but nevertheless it's a critically important element for separating those samples into those known or a priori groups. I'm just going to show you one more example of the application of that tool now. And that's right, Leah's just reminding me there's 10 minutes left, and that's fine, that's plenty of time. Now I'll just open the last demo file now, because this one's a bit silly, a bit of fun. Wine quality, don't save. So I've already done the discriminant projection analysis in this case, but what we're looking at is a database of 1,600 odd Portuguese wines which have been measured for a whole lot of chemical variables related to their composition, and they've been evaluated by a whole lot of wine judges and scored. Now you can see here that here are the wine scores. 3's bad wine, 8's good wine. No one scores things higher than 8. The scoring system goes from 1 to 10. So people typically don't pick very bad or extremely good. They tend to hover around the middle. Now if I turn on the wines that have got middling scores, there's a terrific degree of overlap, but I've turned them off. You can see here that what the judges were pretty confidently able to do was to pick wines that were good, the light blue and the khaki green, from wines that were pretty bad, the light green and the red ones. From a variable importance point of view, it's interesting to go in now and ask the question, well, the judges, when they're tasting the wine, could easily separate them into being good or bad. What in the chemical analysis are most important in allocating those wines into the same buckets? So I have a whole lot of chemical parameters of the wine selected, and if I go into the analysis and discriminant projection analysis and variable importance plot and making an extensive search and run it, you can see here that the 2 most important variables, if you go to the report, for determining whether the wine is good or bad, is the alcohol content and the volatile acidity. And the volatile acidity, if you want another name for that, just call it vinegar. So if the wines are crook, they taste like vinegar, it gets a bad score. And if the wines contain a lot of alcohol, and alcohol tends to give the wines body and warmth, that also ends up being associated with it being a good quality wine. So from a purely analytics point of view, it boils down to whether it's vinegar or has a high content of alcohol. Perhaps a slightly disappointing result. Now I'm just going to jump back into the PowerPoint and wrap up. Go through some, we've covered these. So there's a few other things which I'll just not go into gas, to show you in the PowerPoint. In the principal components analysis now, there's a scree plot. It's a very useful plot. So when people run a principal components, they're often wondering which ones should they look at. This is not super quantitative, it's just a guideline of how many to retain. So when you run a principal components using a correlation matrix, all of the input variables are standardized, so they'll have a standard deviation or variance of 1. This just plots the eigenvalues or the standard deviations of each eigenvalue in turn. And of course, the first one extracted contains most of the info, then less, etc. There's a guideline there at 1. And so what this is telling you, if you're looking at a principal component 5 in that case would actually contain less information than any of the input variables. So it just gives you a guideline about where most of the information is. If you were to run a principal components analysis and this curve was very flattened out like that, it's probably telling you that you don't have many correlations in the input data, and the principal components is really not doing anything for you in a variance compression, from a variance compression point of view. The other thing we've got here is that a lot of people like the ability to export, especially spatial views of data, into Google Earth. Up to GAS 6.2, you've needed the lat-long coordinates in there to do that. If you don't have lat-long, but you have other map system coordinates in the file, then you can go into the map menu, and we can now convert them into WGS84 lat-long. And if you do that, it'll plonk them into the metadata in GAS, and then GAS knows they're there and will be able to create the Google Earth export for you. Where's that stuff? Where's the mapping? Ah, Soph's trying to remind me to say something. So we can now write map info tab files from the 64-bit version of ioGAS. Right. Now that's a big thing. I know it's why Soph's remind me to say that. Some other things here. So if you had workspaces, checkpoints, and templates, sometimes if something wasn't quite working when you were trying to restore it, GAS would throw a wobbly and do nothing. Now we've improved all of that. So GAS typically now in all of those situations will restore what it can, even if all of a sudden it doesn't have a bit of information. It also retains settings between different sessions when you're connecting to a SQL database. There's support now for the ioGAS and Geoscience Analyst, which is a product put out by Myra Geoscience, and by all means talk to the Myra people about that. We have some different tools as well now, so we can double click on a plot window, zooms to the data, you can lock the aspect ratios of plots, and if you right click in there, if you're wondering if you just want to zoom into the space on the plot that a particular color group spans, you can do that as well. If you're looking at the scatter plots and you have multiple colored groups, and you're looking at the regression lines by color group, previously to get the information about the regressions by color group was a bit of a pain because you needed to hover over the plot and write them down. Now there's a button over on the right hand side that just tabulates all of the different regression lines for all of the different XY pairs for all of the different color groups, all in one fell swoop. Some other minor things here, so the legend window now is able to be exported as an EMF or EPS export. You can copy the metadata out of the diagrams that we ship more easily because often that contains reference information, so if you want to put the reference underneath, it makes it much easier. And some little consistency things like hitting the escape key now closes the dialogues everywhere. We mentioned map info on the ArcGIS front. Most of you will know if you're ArcGIS users, we've had a problematic relationship with our Arc export in the past. We're going to wind up the way we're doing it. but right now we're actually working on a separate plugin that will run within ioGAS Pro itself, but we will support it, and that tool will be far more robust and will be able to bring in ioGAS coverages in a very simple way into ArcGIS, and also potentially in the future connect to it in a live way as well. If you're going to go and download GAS or update it to 6.3, always check your maintenance. If you're out of maintenance, then send us an email and we'll sort you out. There's always more information in GAS. Go to the help file, check the FAQ. I always say to people, if it's taking a long time to do something, then you probably missed something along the way, so check those two things. If you're still having a problem, then send us an email on ioGAS support. And for those of you who are local or going to travel, we hope to see you all at the International Association of Mathematical Geology Conference being held in Freo in Perth. We'll be there with bells on. We're a gold sponsor of the event and might even be running the odd thing around it. And then finally, we're always up for running custom training courses on site, which can be handy for companies who want to use their own data as part of that. And our next public training course is listed for the middle of August, so about 6 or 7 weeks away. So if you're interested in, please register. So that's it for now. I'm not sure if there's any questions in the text box. No. I know the seminar's going to time out in a minute anyway, regardless. I think we'll wrap it up there. If you've got any questions, then please just send them through anyway. We've recorded this session, and then at some point in the next few days we'll be putting this up on the website. So if you think someone else would benefit from seeing it, then they'll be able to just go onto the web and have a look. Thanks for your time. Thanks for your attendance. Sorry for the delay at the start. Just some minor IT problems. And good afternoon or good morning, depending where you are.